Can you tell us about your research background and experience?
In my previous role at XYZ Lab, I focused on understanding the molecular mechanisms of cell regeneration. I conducted experiments using CRISPR-Cas9 technology to edit specific genes in mice models. This work helped us identify key pathways that could lead to breakthroughs in regenerative medicine. My findings were published in Journal X, and I presented this research at the National Cell Biology Conference.
What methodologies and techniques are you most familiar with?
I am experienced in using techniques like PCR, gel electrophoresis, Western blotting, and microscopy. I also have extensive experience with data analysis software such as R and MATLAB for analyzing complex biological data.
How do you approach problem solving in your research?
I approach problems by first gathering all available data and reviewing relevant literature to understand the problem's context. I then formulate hypotheses and design experiments to test them. If the results are inconclusive, I revisit the hypothesis, refine the experimental design, and run additional tests. I also regularly consult with colleagues to gain different perspectives on the issue.
How do you handle setbacks or unexpected results in research?
In one of my projects, I was working with a new experimental technique, but initial results were inconsistent. Rather than abandoning the project, I re-evaluated the experimental conditions and consulted with my mentor. We discovered that a small adjustment in the temperature control of the experiment yielded more consistent results. The project was a success, and I learned the importance of perseverance and flexibility in research.
How do you stay current with the latest developments in your field?
I regularly read journals such as Nature and Science to stay informed about the latest research. I also attend conferences and workshops to learn from experts in the field. Additionally, I am part of a research group that discusses recent publications, which helps me keep up with the cutting-edge developments.
Tell us about a time when you worked in a team on a research project.
During a multidisciplinary research project, I collaborated with biochemists, statisticians, and engineers to study the impact of environmental factors on microbial ecosystems. My role was to design and conduct the biological experiments, while also ensuring clear communication with the team to integrate our findings. This collaborative approach allowed us to publish a comprehensive study on the topic.
Why do you want to work with our team? And how do you think you can contribute?
I have been following your company's research on sustainable agriculture, and I admire your innovative approach to addressing food security challenges. My expertise in plant genetics and experience with CRISPR technology would enable me to contribute to your ongoing projects. I'm excited about the potential to apply my skills in such an impactful area.
How do you manage time when working on multiple projects or deadlines?
I use project management tools like Trello and keep detailed schedules to prioritize tasks. I break down each project into smaller milestones and allocate specific times for each task. When working on multiple projects, I ensure that urgent tasks are completed first, while also balancing long-term projects with consistent progress.
How do you handle data analysis and ensure the accuracy of your results?
I use a combination of statistical analysis tools like R and SPSS to analyze data. I always validate the data quality by checking for inconsistencies or outliers before drawing conclusions. Additionally, I conduct repeated experiments to ensure the results are reproducible and statistically significant. I also encourage peer reviews to double-check the analysis process.
Where do you see yourself in 5 years?
In 5 years, I hope to be leading my own research team, contributing to breakthroughs in renewable energy. I aim to publish high-impact research and collaborate with industry leaders to apply scientific discoveries in real-world solutions.
Describe a challenging research problem you've encountered and how you solved it.
While working on a project investigating gene expression in cancer cells, I encountered unexpected variability in the data. I first analyzed the potential sources of error, including sample contamination and incorrect lab procedures. After ruling out these possibilities, I adjusted the experimental conditions, including optimizing reagent concentrations. This helped reduce variability and allowed us to confirm the hypothesis.
How do you prioritize tasks when you have multiple research projects or experiments to manage?
I prioritize my tasks based on deadlines and project importance. I break down larger projects into smaller, manageable tasks and set weekly goals. I use tools like Google Calendar for scheduling and set reminders to keep me on track. Additionally, I assess each project's urgency and communicate with stakeholders if adjustments are needed.
What do you think are the most important qualities of a successful research scientist?
A successful research scientist must be curious and persistent, always asking questions and looking for deeper insights. Attention to detail is crucial, especially when dealing with complex data. Being adaptable and able to work under pressure is also important, as research often comes with unexpected challenges. Finally, clear communication is vital for sharing findings with the team and the wider scientific community.
Can you give an example of how you've worked with cross-disciplinary teams?
I worked on a project that involved collaboration between molecular biologists, chemists, and engineers to develop a new type of biosensor. While my expertise was in molecular biology, I coordinated with the chemists to ensure the reagents worked with the sensor material, and the engineers helped with the design and functionality. This required clear communication and regular meetings to align our goals and ensure the project moved forward smoothly.
What role do you think creativity plays in scientific research?
Creativity is essential in research because the scientific process isn't always linear. Often, you need to think creatively to design experiments, interpret unexpected results, or come up with novel approaches to problems. For instance, when testing a new drug compound, I had to creatively adapt a traditional assay to accommodate the compound's unique characteristics, which led to more accurate results.
How do you ensure reproducibility and validity in your experiments?
Reproducibility is a cornerstone of scientific integrity. I ensure all my experiments are designed with rigorous controls, and I document every step of the process, from sample collection to data analysis, to ensure anyone can replicate the work. I also double-check calculations and run multiple trials to verify the consistency of results.
What experience do you have with publishing scientific papers or contributing to research publications?
I have co-authored several papers published in peer-reviewed journals. My role in these papers ranged from data collection to writing sections of the manuscript. I also worked closely with senior authors to revise and address reviewers' comments. One of the most valuable lessons I learned was the importance of clear, concise writing and being open to feedback.
How do you handle tight deadlines and high-pressure situations?
I stay organized and focused by breaking down larger tasks into smaller, more manageable pieces. I prioritize urgent tasks and set clear milestones to meet deadlines. I also communicate regularly with my team to ensure we're all on track. When under pressure, I make sure to maintain a positive mindset and remember the bigger picture of why the work is important.
What do you enjoy most about conducting research?
What I enjoy most about research is the opportunity to uncover new knowledge and solve complex problems. There's a great sense of fulfillment in being able to contribute to the scientific community and make a real-world impact, whether it's through advancing medical understanding or developing new technologies.
How do you communicate complex scientific concepts to non-scientific audiences?
When communicating complex concepts, I try to break them down into simple, relatable ideas. I use analogies to compare scientific processes to everyday experiences, and I avoid jargon. I also rely on visuals like diagrams or infographics to help illustrate key points. The goal is to make the information accessible without sacrificing accuracy.